Hi everyone, thanks ever so much for joining us this afternoon. My name's Helen Bethel. I'm a registered genetic counsellor and I'm also currently working in the role of Workforce Development and Education Lead for North Eastern Yorkshire Genomic Medicine Service. And this is the first of 3 sessions in the series of ABC of Genomics. So as I said, this is the first session of 3 in a series that I've titled ABC of Genomics, and it's designed to give a really basic overview of genomics. And so the first session today is about the advances in genomics, and I'm hoping to give you all a little bit of context in terms of why genomics is such a hot topic at the moment. The session tomorrow, so at the same time, 3 o'clock, is going to cover the basics of genomics and think about some of those real kind of basic terminology that you might hear, some of the basic inheritance patterns and some of what you do and what you don't need to know when you're using genomics. And finally, the third session on Friday, again at 3 o'clock, is about clinical pathways in genomics. So thinking about how we use that information and those basic knowledge, how do we then introduce it into a pathway that's going to benefit patients? So today's session, the advances, we're going to cover the technology that has driven the changes that we see today, including the introduction of whole genome sequencing, which we often refer to as WGS. We're also going to think about what the NHS vision is for genomics, and I'm going to give you some signposting to some additional learning. We're also going to think briefly about the test directory, and finally also where clinical genetics sits within all of that. So first and foremost, I think the most important thing to start with is the definition between genomics and genetics. So we did a huge roadshow series last year in which we visited over 70% of the acute trusts within NEY. And we had over 2,000 conversations with staff in corridors and in canteens. And we were asking staff if they had heard of genomics, and the vast majority of people answered that they'd never heard of the term genomics. A lot of people had heard of the term genetics, but with genomics, there was less understanding. And so there's been this shift to using genomics because it is a more technically accurate term, but we have a big issue in that people don't necessarily recognise it for what it is. So there is a subtle difference between the two. So when we talk about genetics, we're talking about the study of genes. So they are the exact recipes that are going to encode for the proteins that we need in our bodies to carry out the functions that the body needs of it. When we talk about genomics, we talk about the genes and also everything else in between. So we know that there are certain bits of the DNA that are non-coding, which actually we're learning are just as important as the coding bits in the genes, and also all the other factors which help to regulate it. So when we talk about genomics, we're talking about the whole system altogether, whereas with genetics, we're just talking about the genes. One way of thinking about it is almost a little bit like a book. So with genetics, we're talking about the chapters in the book, whereas with genomics, we're talking about the format of it. We're talking about the structure of the physical book. We're talking about the page numbers, of the spine of the book, of the binding, and things like that. So you can see that the genomics is the whole thing together, whereas genetics is specifically the genes. When it comes to talking to patients and often colleagues, these terms are interchangeable because, again, patients don't necessarily recognise the term either. So genetics and genomics can be used interchangeably. And as much as I am doing my best to use genomics, you might find that I myself might use the odd genetics in this talk today. So where did it all begin? Well, just over 150 years ago, in 1865, Mendel, who was a monk at the time, was studying patterns of inheritance in peas, and he was the first scientist to document that there was predictable patterns in inheritance, and he was looking at whether the peas had wrinkly or smooth skin. A few years later, Misha, a scientist, discovered that there was DNA in the centre of the cell, meaning that he'd worked out that it was critically important to these factors being passed on. In 1955, Tyjo and Leven found that finally pinned down the question of how many chromosomes were in the middle of a human cell, and it was finally accurately described as 46. For several years leading up to that, they had worked out that the chromosomes were the structure that the DNA was contained in, but they weren't quite sure. There was lots of reports of different numbers of chromosomes, but by 1955, the microscopes at the time were then powerful enough for us to be able to detect how many there were. And reliably, these two scientists reported that it was 46. By 1962, Watson, Crick and Franklin were able to identify the actual structure of the DNA, and that's that double helix or that twisted ladder that people will often first picture or bring to mind when we talk about DNA. So that was in 1962. And by 1983, we'd started to work out exactly where these genetic instructions were located within those DNA. So that's the kind of the rapid overview history of the science of genetics or genomics, taking us up to 1983. But I just want to think about that now in the context of health and of testing that we use within a healthcare setting. So back in the 1960s, we were able to recognise chromosomes. So we were able to look down a microscope and see patterns such as this. And you can see here there's a red circle which has identified that this person has got 3 X chromosomes, and we should have either 1 or 2 depending on our sex. This person also has a Y at the bottom, so really there should only be one X chromosome here. So we were able to use this in a clinical setting to be able to look at people's chromosomes and identify that actually this could be the cause of what their symptoms or their condition was. By the 1990s, we were able to start doing direct testing. So this was where we were starting to understand the DNA sequence. So we'd understood the structure since the 50s, and we were starting to understand more about the actual sequence, so those letters in order. And by 1990s, we were able to start picking out slight changes in particular genes and individual genes. If we fast forward to 2015, this was when we started to introduce panel testing. So, for example, given on the screen, kind of breast cancer, where we were able to start testing not BRCA1 on its own and then BRCA2 on its own. We were actually able to start putting these together because the technology could sequence them in parallel at the same time. And by 2020, we had the technology to use whole genome sequencing. So this meant that rather than picking out single genes that we were going to sequence and read and analyse and see if there was a change, instead we could sequence the whole of a person's genome, so from chromosome 1 right the way down to chromosomes 22 and the sex chromosomes. And I'm just going to share with you a little bit of an analogy of the way that I think of this. So essentially, when we're talking about microscopes and looking for chromosomes, essentially it's a bit like a satellite. It's able to look from far away and it's able to see the structure of what's going on. So in this case, we can see from the satellite that there is a lake and there are various ponds around it. By the 1990s, we were able to actually use a fishing rod to actually approach that pond and see whether we could identify a particular type of fish that might be explaining some of the patterns that we were seeing going on. And by 2015, rather than just using one rod at a time, we were able to use multiple rods at a time. And then the analogy for 2020, where we're introducing whole genome sequencing, essentially we're sending out a trawler. So we're sending out a trawler which is going to collect everything in the ocean up. We're then going to analyse what is in our net. So getting rid of all the usual things that we might recognise, so the jellyfish, so the dolphins, the crabs, anything that looks typically normal, we can get rid of that. And then we can look to see what else is still left that might be different, that we might not have seen before, or we might recognise as similar to other patients with a similar presentation. So whole genome sequencing first came onto the scene in 2001 when the first draft of the human genome was completed and published in this paper. It was an international effort by the Human Genome. Oh, I think someone's just unmuted there. Can you just make sure you're on mute for me?
Spread the word.
To me, it means like
I think. Thank you, Laura. Thank you. If you can just make sure you stay on mute, that'd be great. So by 2001, the International Human Genome Sequencing Consortium had managed to sequence from chromosome 1 right to the end of the genome sequence, so chromosome 22 and 23. They had managed to read all of that data. They hadn't done any analysis of it, but they were literally able to read it with the technology that we had. It had taken them 13 years and it had taken them over $100 million to complete it. And obviously with this timescale and with this price, it's not feasible to see that fitting in any healthcare system. But if we look what happens over the next 20 years, I think this slide is really key. So this slide shows us the cost on the left-hand side from 2001, where the cost of a human genome, one single human genome, had cost that price of $100 million. The white line shows us the predicted cost of that technology over the next 20 years. And the green line shows us the actual cost. So it shows us that by 2001, the cost of carrying out this test of sequencing one person's whole genome, the cost was around about £1,000. And suddenly it's at that level where it becomes interesting, because it's at that level that we can maybe start considering introducing it into a healthcare system. So fast forwarding now to more recent history, if we look from 2012, we've got the introduction of the 100,000 Genome Project. So this was a feasibility study, which was part funded by the government and was essentially looking at, can we feasibly carry out whole genome sequencing on a large scale for a group of patients where we don't yet have a diagnosis, but we suspect that there is something genetic going on? Is it possible to do that, and is it acceptable to patients, and is there demand for it? So in 2012, the 100,000 Genome Project was announced, and by 2014, we were recruiting patients into that, and by a year later, some of those diagnoses were then returned. We hit the mark of sequencing 100,000 genomes in 2018, although, as I said earlier, there is a difference between sequencing and analysing and producing results. So although we have sequenced these many genomes, we haven't necessarily reported all of the results, and so analysis is still ongoing. By 2022, the government then published its strategy of accelerating genomic medicine in the NHS, where it outlined its real ambitious target of being the first national health service to offer the whole genome sequencing as standard for and embed it into patient pathways across the NHS. Because it was massively upscaling the amount of genomic testing that was being done, this was at this point was then recognised that this was far too much to be offered in a small specialty like clinical genetics. So it was decided that that testing was actually best placed for the patient to be within their pathway, and that's what brings us on to the mainstream agenda that we see today. So if we just look at this slide in terms of the vision for genomic medicine within the NHS, if we look at the top, we've got a traditional model. So in a traditional model, a patient would be seen with a health complaint, a diagnosis would be, or a clinical diagnosis would be made. There may be red flags, or there may be family history that flagged the presentation that actually indicated that there might be something genetic going on. And if it was, then that patient would be referred through to clinical genetic, where they would have their family history assessed, and they would access testing if it was appropriate. If once testing was complete, whether diagnosis was confirmed or not, and then there would be a referral back to mainstream medicine for their management. In the mainstreaming pathway, essentially this step to clinical genetics for diagnostic work is taken out. So essentially the diagnostic work is done by that initial mainstream team. So if there are red flags, if there is family history or presentation that indicates that there might be an underlying genomic basis to the presentation, then testing can be offered within a mainstream department. Results are then applied to help make decisions around treatment and management. And finally, if there is a genetic result and it has implications for other relatives, then the patients are still referred into clinical genetics, so that clinical genetics can take that role in still cascading testing for other relatives, and also family history assessment is required, because obviously there can be negative people who might still have, although they don't have a specific genetic diagnosis, there may be still an element of genetic risk, and so there may still be screening and management for family relatives. So there is still a role, even in negative results sometimes, for clinical genetics. So a key part to this process of enabling those mainstream pathways to prosper is the test directory. So in the traditional model, as I say, all genomic testing had to be requested from clinical genetics, but in the current mainstreaming model, the idea is that clinicians can request that testing if it's relevant to their specialty. So the test directory is a document which is regularly reviewed and updated, and it's available if you just pop it into Google, or there is a QR code on screen if you prefer. The test directory is basically outlines the eligibility criteria for all the tests that are commissioned within the NHS. So prior to this, when clinical genetics were carrying out a lot of the genetic testing, there was a lot of inequity around the country because testing was done in different ways in different labs. So the test directory standardizes that. So it tells you exactly what the eligibility criteria is. So it doesn't matter which part of the country you are living in. This does only apply to England and not the devolved nations, I must say. So it gives you the eligibility criteria, and it tells you the specialties that are able to request that testing. So if you sit within that specialty, if that patient meets that criteria, then you can request that testing for that patient. So I've already referred a little bit to this, but let's just think for a moment about where genomic medicine is in this new world. So new pathways are that I've described here are very much around the diagnostic testing only. So we're not asking clinicians to start testing people who are unaffected or to start taking responsibility for cascading that information. So genomics is very different from other types of medicine in that in the majority of medicine, when you make a diagnosis, it's usually only relevant to the person or the patient who's sat in front of you. Whereas in genomics, obviously, we have a whole family element to consider. And this is where we really are specialists and where, we've got experience of many, many decades. So it's a bit like a drop in a pond. You deal with the drops in mainstream, but the ripples are for clinical genetics to pick up. So where diagnoses are made, then patients can be referred into clinical genetics for support around that diagnosis, and also support in helping to tell their relatives, and then for helping to test or cascade people predictively in the family who might also want testing. Clinical genetics is also there to help support. So if you are finding that you have a particularly difficult issue, if there's an ethical issue that's cropped up, or if you're just wanting advice and support around maybe some of the issues that have cropped up in the conversation with the patient, then clinical genetics is still there. And very often clinical genetics are taking part in multiple MDTs, in multiple specialties who are introducing genetic testing so that we can help support clinicians through that. So please do reach out to your clinical genetics service if you're stuck. So if you found today's session engaging and you would like to know more, then you could consider following NEY Genomics on social media via LinkedIn, Twitter or X. And there's also a QR code there to sign up to our newsletter. You could also consider coming along to our next session, the basics of genomics, and finally on the third session, the clinical pathways in genomics. We also have a lunch and learn series, the next of which is also tomorrow. So busy week this week, which is on inherited breast cancer. And I'd also really encourage you to have a look at these two websites that are listed here. So the Genomics Education Programme has got a whole host of resources, right the way from bite-sized videos up to master's programmes, and also GNotes. So GNotes is a just-in-time clinical resource. It's in two parts. It's got a knowledge encyclopedia, and it's also got an in-clinic arm. And the in-clinic arm offers lots of different scenarios for lots of different specialties to help support and direct people to learning and resources that might be of use in various different clinical scenarios. If you want to do some more formal learning, then again, you could take a look at our website or again at the Genomics Education webpage. So I'm going to stop sharing in a moment so that we can do a question and answer session if anybody's got them. But it's just to say that we do offer a certificate of attendance for each of these sessions. So if you would like to obtain one of those, you just need to fill in a very quick evaluation form that takes about 2 or 3 minutes to fill in. The QR code is on screen, or alternatively, I'll copy and paste the link into the chat for you in a moment. Thanks so much for listening.